gonna stop it. So let's see if this is working. No, that's working.
I can just advance with the arrows.
See, it should be working. Sometimes you have to do it twice. And what is this? The arrows go back? Yeah.
I'll just do that.
Oh no, here it is.
Oh, cool. Alright.
Turn the other light on.
Cool.
You want this light on or off?
Either works. Yeah. They can let me know. Actually, can you see the screen pretty well with the lights on?
There's
Okay.
on, there's off.
That's better.
Off? Alright, let me put you all to sleep. Oh. Hi, welcome. I want to thank Math Club for inviting me. This is a cool experience. You also set the bar pretty high. The flyer said I was going to give an exciting talk about bioinformatics, so hopefully that measures up. We'll see. Hi, so I'm Amanda Petrus. I teach chemistry here. And I am a biochemist, and some of my research projects have overlapped with areas of bioinformatics. So I'm here to talk to you about this area of science where a whole bunch of fields, I can't even say science, it's math, it's science, it's everything. So this is a field that computationally analyzes a lot of data that's biological in origin. And we're combining computer science, engineering, biology, chemistry, mathematics, medicine. All these different fields converge into one when you look at bioinformatics. And if you're a bioinformatician, which is a hard word for me to say, you're very flexible and applicable to a bunch of different fields. So in a way, it's a powerful skill set to have. And there's even an artistic side to this because it's a very visual field. A good bioinformatician takes a huge amount of data, a terrifying amount of data. They look for patterns in it, they find a story, and then they make a simple figure that expresses that idea in a clear way. So there are a couple examples here of different ways that bioinformaticians can express data. This is something called a gene heat map that I'll talk about in several slides. But all these color changes are different data points, and you can see it's a phenomenal amount of data in one figure. This is a figure of a genome showing a whole bunch of gene transfers and different gene events happening, and this is kind of a fancy way of looking at an evolutionary tree. So all these different things come into very visual form of science. All right, so it's all about this data. When we talk about big data, what kinds of data do bioinformaticians work with? Protein sequences and DNA sequences. You know, we are in the era of the completely sequenced genome. We've had the human genome completed for several years. So we can take all of this information that's loaded into databases, and we can analyze whatever we want. So you can get information about protein structures, gene expression, the identities of genes, the roles that DNA play. You can predict how two proteins will interact with each other. You can predict how a drug will interact with a target molecule, target receptor, so you can kind of predict how something might act biologically. You could do genome-wide studies where, let's say that you're interested in looking at a certain population with a medical disorder. You can sequence just thousands of people's DNA and look for patterns with that disorder in certain subpopulations. You can look at evolution, and you can track the changes in DNA over time to figure out how certain changes have happened in organisms on a DNA level. You can watch evolution happen in terms of an organism, or you can see it happen on a smaller level in terms of DNA patterns. And epidemiology, this actually has a really cool application for tracking disease outbreaks. We've been dealing with things the past few years like Ebola, Zika, all these terrifying things. And there are scientists that are able to sequence samples of viruses and bacteria that have been causing trouble, and they can actually track the source of origin, like the geographic area of the world that the strain of Ebola came from that started the outbreak, right? Some other really cool studies have found an exact area in Africa of a few square miles where the first incidence of HIV transmission between monkey to human occurred, right? And this happens with people just churning through genomes, analyzing data, looking for patterns, and following the story that they tell. So all of this data analysis, since we're dealing with huge data, it's impossible to do by hand, right? You wouldn't be able to get through a lot of this data in your lifetime. So you need computer programs and algorithms to churn through a lot of this data for you. So these algorithms are based on graph theory, artificial intelligence, soft computing, data mining, image processing, and computer simulations. So you've got a variety of different aspects of computer science coming together. And those algorithms are based on some fundamentals of math and statistics. They're based in discrete mathematics, control theory, information theory, systems theory, and statistics. I would say out of all of these, statistics is the one that comes up the most in bioinformatics discussions, because we often talk about probability, maximum likelihood, that kind of thing. And I do have to give a disclaimer as I talk about this. I'm a biochemist by training. Math, we've come to an agreement, math and I, So in terms of, I could talk all day about the biological applications, right? But in terms of the mathematic end, a really deep discussion, I could probably learn a lot from Math Club about how these things are applied to the field. So let's look at specific applications. We've talked about what bioinformatics is and the fields that it looks at. One of the first major applications is sequence analysis. So this is where we're looking at genomes and genes and even proteins. And we have all of these sequenced organisms that have been uploaded into databases. So whenever you see a paper, new bacteria discovered, genome published, that genome that they've sequenced has been uploaded to a free database that you can go online and access. And you can get that whole genome. So scientists have all of this data at their fingertips. Even students that are just interested in looking and finding patterns themselves, you don't need to be in a research lab to access this data. It's all out there. So you can take these sequences and you can look for information that the genome tells you. The genome tells you a lot about what kinds of proteins it will encode, what kinds of RNA it will encode, and it can even tell you a bit about regulatory methods and the structures of things that are created from this DNA. So you can also compare differences across genomes. You can have two very different organisms that have very similar proteins in them that do very similar jobs, right? This is an aspect of evolution. We see things branching off and we see evolutionary pathways changing, but a lot of very, very different organisms have very similar proteins doing very similar jobs, right? A really simple level of sequence analysis, if you had to do sequence analysis by hand, you could do a single alignment. And what that is you take a protein that you're interested in, and that's the top one. And proteins are really long, so this is up to like 330 amino acids. So the top row is a protein that we're interested in studying. The bottom row is a known protein that we're aligning it with. And you basically line them up and you see what portions of the proteins overlap, where do they have the same amino acids. And those areas of similarity are areas where you can kind of say that these proteins are related, right? And the more overlapping areas, the more closely related these proteins are. So that's what you can do visually, right? But if you're working with a maybe 100,000 proteins that, one genome can encode thousands and thousands and thousands of proteins. So if you're looking at a whole bunch of proteins at once, you can't sit down and do this by hand, right? So this is where these computer programs come in. And there are two kinds of There are two kinds of programs I use with quotations for bioinformatics. There are the bioinformaticians that write the code from the ground up, and these are people that have experience in Perl, Python, C++ languages, and they build these programs to do exactly what they want. And then there are other people that take already created programs, load their data into it, and then use those for the purpose of their research. So if this is something that interested you, you could come from either way. You could approach it as a programmer that can write everything from the ground up and be very specific, or you can come at it as a biologist or a chemist that knows enough of the computers to just kind of find what you need to use and use it. I have to admit I'm a little more of the latter than the former, but I have a colleague that is one of those ground up programmers, and she writes some phenomenal code. So one of the easiest things for everybody entering bioinformatics to use is a program called BLAST, Basic Local Alignment Search Tool. And this is like the entry program to look at sequence alignment. You can find anything that you have a sequence for, you can compare it to all known sequenced organisms out there, and you can find what it matches, what it's similar to. This is a way to either identify an unknown protein, like let's say that we isolate a protein from an organism and we wonder what it is, we have no idea. We can sequence that protein, and then we can put that sequence into the BLAST database, get a match, and find what its probable identity is, right? Or if you're interested in evolution and looking at, like one of the things I'm interested in is a certain enzyme called cobalamin reductase, works on vitamin B12, and I'm interested in how different organisms have changed over time and how maybe a lactobacillus E. coli reductase would be related to like a thermatoga thermatoga reductase. So you can do that for evolution, for identification, all that stuff. And this depends highly on probability and statistics. So you get matches, but it's a way where you're looking at probability, and there's a certain score that these matches have to make. So if it's a very good match, it gets a score higher than a threshold value you're looking at. And this program will only give you back scores that hit that threshold or above, right? So I'm going to show you how to use BLAST just once. But as I was saying earlier, you can identify species, you can predict the function of protein, you can look at evolutionary relationships, all that kind of stuff. So let's talk about using BLAST. Since there are different sequences, you can have a protein sequence, or you can have a DNA sequence, or you can have an RNA sequence. There are different BLASTs to use for proteins and DNA and RNA. So the first thing you ask yourself is, do I want to analyze a protein, a DNA sequence, or what? So I'm going to show you using a protein. So blasting a protein, you open the BLAST-P program, BLAST-P for protein, and you obtain something called a FASTA file online, and it's a specific formatting where it's got a little header and then it's got the amino acid sequence, and then you paste that right into BLAST, and then you just click BLAST. It's brilliant. So this is the screenshot of the BLAST website, and if you select BLAST-P, this is where you put your sequence in. And when I say FASTA file, you see you have this little caret and then a GI number and then a name, and you start to see the amino acid sequence. So you paste into that window, and then if you have specific things that you need to watch for your experiment, you click specific things. But generally, you can just select BLAST-P and then hit BLAST. And you wait a while, and after several minutes, depending on how long your protein is, you get this feed. And what this feed is, on the top, it tells you if there's any recognizable domains in your protein. And a domain is a known structural motif that's linked to some kind of function. So if there's a protein that binds metal, there's usually a specific metal binding domain in that protein. And tons of these domains are characterized and cataloged. And so the first thing it tells you is if there's any domain or family identification in your protein. That can help you for identification. Then down here are the top BLAST hits. And this is the sequence, like the first amino acid up to, I picked about a 600 amino acid protein to blast. And these are the top matches. You'll notice a lot of them have like 100% match, and then some of them have missing areas. Missing areas, white, are where they don't line up at all. The sequences are too different. And then the pinkish areas are where they match, but it's not a really high, you can't trust it, right? So below that, the next thing you see is, and we can't read this unfortunately, but each one of these is one of those red lines that was on the previous screen. So here you can go and you can see the exact protein from the exact organism, the exact species. And then over here is statistical and probability information on how much you can trust that match. Yeah.
I'm sorry, may I interrupt?
Mm-hmm.
What do you actually start with? Somewhere there has to be a physical thing which contains that protein.
Yep.
What's been done to that?
So like, how do you isolate the protein? Is that
That's been done somewhere.
Yep.
And it's been converted into a form, the information's been converted into a form that can be accepted by this. Is that?
Yes, that's correct.
Am I
Yep.
understanding correctly?
So you can come at this two different ways. You could be an experimenter in the lab. They call it wet lab and dry lab. So if you're actually growing things in the lab, mixing things, it's called wet lab. And if you're just doing computational analysis, it's called dry lab. So if you're coming at it from a wet lab approach, let's say you would grow an organism, let's say it's a bacteria, and you would harvest those cells, you'd break them open, and you'd isolate the protein you're interested in. Then you would sequence that protein. Usually you have to do certain chemical reactions and get it sequenced. And then once you've got that sequence, you know the exact amino acids in it, then you can feed that into this protein. If you're totally a dry lab approach, you would just say, I am interested in this organism, this species, and I'm going to find its genome online, and I'm going to go download all the genome data for that organism, and now I'm just going to play around with copying and pasting and putting into these programs, or writing my own programs to do really specific things. So you can come from either side. Yeah. Yeah. And actually, if you are someone that can do both, if you can do both wet lab and dry lab, it makes you really valuable. And it often confuses people because most people don't realize people can do both, and they're like, wait, what are you about? So it's rare and it makes you valuable. So if we could see these numbers clearly, you'd see that there's an identity percentage, and that's basically how much this protein matched, how much the protein that you're analyzing, that you fed into this program, what percentage of it matches the proteins listed here. And it's 100%, 90-something%, we get down to 80%. So as you see, as you go down, they post the less likely matches. And then the E-value is very important because the closer the E-value is to 0, the more trustworthy this match is. So we've got a lot of zeros, and you'll notice maybe the first 20 matches have an E-value of 0, and then we start to get 2 times 10 to the 176th. So usually your first big chunk, and this kept going, I just took one screenshot, but you could have hundreds of different proteins to look at. And the further down you get, the worse the match is, right? So from this point, you can pick things to look at. You can say, like, okay, it's really closely related to these proteins, so I'm going to look at these proteins more and try to draw relationships between the one I'm interested in and the one they match. Or you could go from this point and start to make a phylogenetic tree to see the evolutionary relationships to things. So you can do a variety of things. BLAST is usually a starting point. You blast something and then you figure out what the rest of your experiment is. And yeah, as we go, if there are questions, feel free to ask as we go. So BLAST is like one of the first programs that a bioinformatician gets to know. Another useful application for bioinformatics is genome annotation. So if you have a brand new organism that's never been sequenced before, never been identified, you can take that organism, sequence its genome, and then use computer programs to annotate the genome. Because the genes are broken up by very distinct start codons and stop codons. And when you see when you have a start codon encoded into the genome, it tells the machinery making all the proteins, start making a protein now, right? And then it has a stop codon saying, stop making the protein. So there are programs that can go through and actually label all the start and stop codons for you. And then from that, you can get an idea of how many genes are in an organism, how big some of the genes are, And then you can begin to analyze individual genes and see what they all do. Evolutionary biology, here's another major application. I've spoken about it briefly already, saying that you can take data and make phylogenetic trees out of it. Phylogeny is a specific term for looking at evolution in terms of statistically relevant relationships and drawing like trees, so to speak, based on this. So you can trace evolution through individual changes in DNA. You could compare entire genomes, or you could just say, I'm interested in one gene, right? Maybe people are interested in, Ebola was a huge problem a while ago, and there were specific surface proteins that were very important in how Ebola affected the cell. So there were people out there specifically looking at like the evolution of those surface proteins and analyzing them and seeing how the proteins changed as the bug evolved and got smarter and stronger. So there's some cool patterns that you can see if you're looking at genomes. You can see gene duplication, where sometimes extra copies are made. You can see something really funky called horizontal gene transfer, which is where two separate species actually share DNA. Instead of having a linear relationship of DNA, like parent to organism, parent to child, it's actually like two totally different species that are swapping DNA. And as we've gotten better at analyzing genomes, we're seeing that this happens everywhere. So if you look at the tree of life, there are huge chunks of some organism's genome that have come from another branch of the tree of life. It was really controversial for a while, up until a few years ago, but now enough people are studying it and enough people have proven it. And then you can also look at speciation. So sometimes in these genome analyses, you realize that what you thought was one species was actually two different species. They differ enough to be a completely different group. And you can just look at a huge number of organisms, and you can even assemble a tree of life. So this is the tree of life that a lot of us are used to seeing from biology classes. We begin with the most recent common ancestor coming up out of the primordial ooze, and through all of these branching fungus and dinosaurs and mammals and humans and politicians, they all come out on their different sides, right? Phylogenetic trees, they look a lot different, right? Blah. This is how a bioinformatician sees the tree of life. And if you break it down, you can still see that there's bacteria, there's archaea, there's fungi, there's humans, but they're looking at it in different ways. And they can zoom in on all these individual places, and they can look at smaller branches, right? So this is one way that they look at it. And the common ancestor is in the center here in this diagram. Actually, at this point here where it comes up and branches out. And then here's yet another even more terrifying tree of life. So the trees get really complex, right? So to save space, and this is what I mean by kind of the artistic side of bioinformatics, instead of having this in a linear function and probably having it take up like 5 times as much space as it is, bioinformaticians have turned it into a circular thing where the most recent common ancestor is in the center, and then you have things branching out as you go on. And you can see that we've got animals, plants, fungi, all this stuff, and you can zoom in on different areas, right? And everything means something in these kinds of diagrams. Even the lengths of the branches mean something. They can be related to how closely things are related, and then every one of these branches has values to it that express this is how statistically likely this event is, right? Also, what's interesting about bioinformatics is you can kind of do it any way and get different results. So there's many different programs to do these things, and you can even write your own programs to tweak the parameters you're looking at. So you can get vastly different results depending on what parameters you put in, right? So it's a field where you really have to be careful when you're comparing 2 different people's studies, because those 2 different people may have had completely different thresholds. They may have had completely different parameters that they're looking at. So it's a powerful field, but at the same time, you do have to be very careful that people aren't falsifying data to say what they want it to say. And then from a human standpoint, and I've talked a lot about bacteria and all of that, but these have huge applications in terms of disease therapy, right? We can look at genome-wide associations with specific kinds of diseases, profile genomes with diabetes, Alzheimer's, Parkinson's, all these different kinds of things. And we can learn a lot about the genetic origins of diseases and even predict diseases. This is huge for cancer, right? We've heard a lot of people finding out they have the BRCA1, BRCA2 cancer gene, right? Their genome is sequenced, and then someone looks at that genome and identifies those specific genes that increases their risk for cancer. You can also track specific mutations in specific strains of cancer, and you can learn how cancer changes over time. You can also tailor treatments to different strains of cancer. So this is where we get bigger and bigger and bigger in terms of all the possible variables, because not only are we looking at just the probability of association, this gene means this disease, right? Now we're getting to a bigger level where we've got organisms that are changing over time and different lifestyle factors, all of this. So when you get to this level of bioinformatics in a medical field, you have to think of a lot of different factors, right? Let's see. We get huge amounts of data. So in one cancer study, you can get terabytes of data from one experiment. So like I said, it's impossible to sit down with a legal pad and a pencil and go through all this stuff. You really need a lot of computer power, a lot of churning, a lot of churning power, right? And a lot of this data can have noise. So you get data, but you also get a lot of noise from it. And there are a lot of models that help to pull the data out of the noise and take care of that signal to noise ratio. And then there's another thing. Depending on what model you use to filter out noise, you can get completely different results. Yeah.
What does noise mean in this?
Noise would mean, let's see.
Is it just like extra information, or is it like information that actually didn't come from?
It can be extra information. It can be contamination. Maybe you're sequencing an organism, but you've also got human DNA in it, and you've got some yeast DNA in it because things have just fallen into the sample. It can also be they're finding the most probable outcome when they're giving this data, so there might be multiple outcomes. So you might get, like 3 or 4 possibilities, and then from that you've got to pull out the most probable. So some of the less likely possibilities can also be considered noise. Yeah.
So the computation programs automatically distinguish between the needed data and the contaminated data?
Yep, they can. And then depending on how that program's written and what the limits are set at, it'll pull out different things. Yeah. And that's something, if you're someone that can write your own programs, you've got a lot of power to really pinpoint exactly what you need to pull out of that. We can also look at gene and protein expression and regulation. So besides just the identity of a gene, we can look at how genes are turned on and turned off, right? Which can have a big application with diseases, with development, all kinds of things. So you can look at gene expression by measuring RNA levels, or you can also isolate proteins, run protein microarrays, and do something called mass spectrometry. Mass spectrometry is a technique that helps you sequence a whole lot of proteins or a whole lot of DNA at once. And then from these microarrays and this mass spec data, you can compare it to databases, and you can see what other kind of regulatory events give patterns like this, right? Let's see. This is also another really noisy method, so you've got to take a lot into account when you're working on that stuff. Here's that heat map that I talked about. So heat maps are a way of expressing, let me pick a different word than expressing, a way of showing gene expression, right? So in this case, green means go, red means stop, and we're looking at how these are many, many different genes throughout this whole thing, and we're looking at how they are either turned on or turned off under a certain set of stressors. Right? So this is a way of looking at either it's looking at the genomes of several different organisms, or it's looking at one organism and many different genes in different conditions, right? So every heat map is different. It depends on the parameters of the experiment, but it's generally showing what genes turn on, what genes turn off, what intensity do they get regulated, and under what conditions, right? So this is just, if we look at this here, this is a phylogenetic tree on the side. So you can see there's a really dense amount of like tons of different, either these could be like different proteins or they could be different organisms, again, depending on what your experiment's doing. And you can just see, it's a terrifying amount of data, right? So to be able to create something like this that lets you see patterns quickly, that's power. Another really cool application, and I have a real soft spot for this because I really like to think about proteins in 3D, is predicting structure, right? So amino acids have very specific properties, and if you look at a protein sequence, you can determine like, okay, proteins that have these kinds of amino acids tend to be this shape. If things are put together in this order, they tend to have this function and look this way. So you can have computer programs that analyze the order that these amino acids are put in, and it can predict what the three-dimensional structure is when these things kind of assemble in biology. So this also shows that proteins with really different sequences can have really similar structures. So you could have proteins that have totally different amino acids, but the amino acids are kind of close enough in behavior so that they are actually, you've got two proteins, completely different sequences, but they fold into the same shape and they have the same function. And one of the examples of that is many species have a different version of hemoglobin, and a lot of the sequences can be very, very different, but all of those hemoglobins generally have the same shape, do the same function, and carry oxygen through the blood, right? So these structural studies, they rely on a specific aspect called molecular dynamics, where you're not only simulating the most likely shape that something will have, but it's got movement in it too. So if there are multiple objects in the system, you can consider how every individual atom might be moving over a certain amount of time. So not only can you get a picture, but you can also get an idea of mechanisms and how certain things might act. Again, this relies a lot on math, physics, probability, and this lets us look at like the atomic scale of how things might work. We can look at 3D structures, we can look at folding patterns, meaning how does a protein go from that floppy linear shape into a useful three-dimensional folded shape. And it can also help us target drug interactions. Like let's say we're interested in a drug that binds to the estrogen receptor of this strain of cancer cells, right? So we can look at the estrogen receptors on that cancer cell, model it, and then we can model our drug and see, does it bind? Does it block the receptor? Is this a good drug candidate? So it's really cool, and it ends up making really gorgeous computer images. So those are the main areas of bioinformatics coming from my standpoint, coming from a biochemist that enters things. Now what I want to talk about is two different projects that I've done so far in my career that use bioinformatics in two different ways. The first one I'm going to look at is using drug delivery, using vitamin B12 to deliver insulin orally. And this is a project that uses the molecular modeling, the molecular dynamics. And then the second project I'm going to talk about is biosynthesis of vitamin B12 by bacteria. And this takes into account looking at different protein sequences and genetic sequences, making phylogenetic trees, and figuring out an evolutionary story based on big data. So first off, vitamin B12. If anybody's been cornered by me, you know that I love vitamin B12. It's borders on an obsession. It's a vitamin, and it's the most complicated vitamin that we have. Most vitamins are only like a quarter of this size, right? Vitamin B12 is huge. It's got a lot going on, and it's the only vitamin that has a metal in the middle of it. This cobalt in the very center. This makes it the only organometallic vitamin. It's essential for a lot of proteins to do their job. So a lot of enzymes that we use to stay alive need this vitamin to do a lot of different jobs, right? So here's just a flat stick drawing that you would run into in organic chemistry. And then here's a ball and stick 3-dimensional model. So I just want to point out there's a cobalt in the middle. There's a ring here called a corin ring. And then there's a bunch of stuff on the outside. So as I talk about that, just keep those things in mind. So, Project 1, oral delivery of insulin. So, insulin is a way to, supplying insulin transdermally is a way to treat diabetes, right? The problem is injecting insulin is painful, it's time consuming, it's also pricey to always have the needles, the clean needles too. So, if we could administer insulin orally, it would be much easier, much nicer. Who would rather take a pill than stab themselves, right? The problem is insulin is a protein, and just like when you eat a steak and that goes into your stomach, it gets ripped up and chewed up and totally broken down. So insulin can't get through your stomach to go into your bloodstream to actually work on the blood glucose. But vitamin B12 can go through your stomach, and it can get carried into your bloodstream. And in fact, it's such an important molecule that our bodies have developed a transport system for it. There are a series of proteins. The very first one comes out in our saliva. So when we eat yogurt, yogurt's a great source of B12. You eat yogurt, there's a protein in your saliva that immediately wraps around B12 and protects it and carries it into the stomach. And that protein wraps around it, protects it from the stomach acid. It goes into the intestines, and that protein hands the B12 off to another protein. And then that protein hands it right to the intestinal wall where a receptor takes it into the bloodstream and then releases it. And then there's a protein that carries it through the blood. So there's this great shuttle system that the body has worked out. And when my boss hired me to do my PhD, he said, what if we took vitamin B12, which is this guy here, and what if we actually covalently stuck insulin to it here? So what we did was we made a new chemical bond. We attached vitamin B12 to insulin, and we asked, can insulin sneak along for the ride with these transport proteins? And it actually did. So we found that the B12 system could handle the insulin. The insulin got through the stomach into the bloodstream in diabetic rats, and it actually lowered blood sugar by about 30% the effect that an insulin shot would have, which was a pretty good start. So people that took that project over after I graduated are working on making it better. But when we synthesized it and we proved it in a wet lab manner, we then asked ourselves, is there anything that we can do in a dry lab manner to support our project, right? So a collaborator went on to make a bunch of images for us, and he made this beautiful image predicting that there was B12 and there was insulin and there's a very flexible tether here, and that flexible tether is part of the way that it works. And he made some images that showed vitamin B12 and insulin sticking to the B12 transport proteins. So this yellow unit right here is the vitamin B12, and this ribbon structure is the B12 transport protein, and that tether attached to insulin sticks right here, and the insulin actually fits in that protein and gets carried along for the ride, right? So we found out that those transport systems worked. It was great. The B12 attached to insulin works with the B12 system. And then we went another step further and we asked, can the B12 insulin actually interact with the insulin receptor? And this one was a gorgeous picture. So this is a modeling system that predicts the probability where every single atom could be in the insulin receptor. And this butterfly-shaped structure is the insulin receptor. Picture this being in a membrane right here, right? And if we look at this, the red in the very center is the B12. This aqua-colored structure is a B12 transport protein. And what you can't see, it's blocked, but the insulin protein itself fits and is able to dock exactly where insulin is supposed to go. So we proved it on a physiological level. We saw a reduction in blood sugar, and then we proved it on a computational level, showing everything fits, right? So this was a fun project, and I enjoyed seeing everything from start to finish, you know, actually making something all the way to seeing the final result. So that was the first project. And then the last one, just check my time, okay. The last one was looking at vitamin B12 and how bacteria make it. So we have to get vitamin B12 from our food. We cannot make it. Bacteria and archaea are the only things in that huge tree of life that can actually make vitamin B12. So we get it from anything that bacteria grows on: yogurt, cheese, meat, all that stuff. So when I went to UConn and I did my first postdoc, there was somebody there that was working on vitamin B12 and bacteria, and they said, "Oh, you should join us because you actually know what this big molecule is and it terrifies us." And I said, "Okay, phylogeny terrifies me." And they're like, "That's okay, we'll all get through this together." And I was like, "Awesome." So we learned a lot from each other. So we looked at a certain species called the thermatogae, and the thermatogae are a heat-loving organism, a thermophile, and they can grow anywhere from 37 degrees Celsius to like 80 degrees Celsius, right? They're really, some of the cultures I was growing, it like hurt to hold onto the bottle. It was too hot, but they were happy. So we found that they can make vitamin B12, and we found through scanning the genomes that they had really unique organization of they not only had B12 genes, but they had them organized in really unique ways. So we also found that a lot of these genes were also present in distant organisms on other branches of the tree. So we found that they shared a lot of really similar things to firmicutes and archaea. So we went on to look that on a wet lab sense, we actually grew the bacteria in vitamin B12 free media, and then we analyzed, after they grew it, we analyzed it for vitamin B12, and we found that they made B12, right? So we proved on a wet lab way that they made B12, and then from a gene way, we looked at, they have all the genes, and we actually constructed an interesting evolutionary story about how they got the genes. So here's a simpler version of the tree of life. It's a little less intimidating than that giant circle thing. And here we've got the most recent ancestor, and we've got bacteria branching off, we've got archaea branching off, and then all of us. So the Thermatoga are here, and they're really close to that most recent common ancestor, and that makes them a ancient bacteria. So the things that are closer to this base of the tree of life are considered ancient. Their genomes are they're unchanged, more unchanged than things that are further up. So it was kind of interesting. It was like time traveling, going back in time and seeing the origins of this vitamin B12 molecule. And we found that there were a lot of genes that it shared with Aquifex. So they had some similarities here, and some genes they shared with archaea. So we thought that was interesting, and we thought, horizontal gene transfer? Maybe. Let's see if we can prove that. So I want to point out it's a complicated molecule, and it's a really complicated synthesis. If we were humans in the lab, it would take about 70 steps for an organic chemist to make it from start to finish. One guy did that. It took him over a decade. He had 100 people working for him. So actually 100 other people did it, and he took the credit. But it took him a decade. It took him 100 coworkers, and he got a Nobel Prize for it. He did it in 70-some steps. Bacteria can do it in 33, right? And they figured it out like 3 billion years ago, so they're a lot smarter than us. So there's 3 ways that they can make B12. Well, there's 3 ways they can get B12. The first way is called de novo synthesis, where they make it from scratch. They start with a tiny little molecule, glutamate, and they build a giant B12 molecule from it. The next way is they can take incomplete, coronoid is a term for that ring structure. So they take incomplete coronoids, these are called cobinamides, and they finish making them. So these bacteria or organisms, they've got like half the genes, and they just take somebody else's work and they finish it, right? And then the last way is they can't make it at all, and they just steal from other people, and they use a bacteria, They use a transporter to bring it in. So here's something from a metabolism database, and this, if you're really into metabolism and bioinformatics, check out a site called KEGG, K-E-G-G. This is from the KEGG database. This is the biosynthetic pathway for vitamin B12, and we start here with glutamate, and every one of these dots is a new compound. It gets changed along the way, and every one of these boxes is an enzyme that does that job. So you can follow the synthesis of B12 from glutamate all the way down here to the finished product, vitamin B12. And just an interesting note, there are some other things that kind of look like B12, like the corin ring looks a lot like a heme ring from hemoglobin, and heme and corin actually share all these genes, and then heme goes off this way if you want to see it differentiates and goes off in this direction. So, but for this purpose, we're just looking at this part of the diagram right here. So, de novo synthesis shows that, from glutamate to vitamin B12. The second way is some of those organisms only have these last genes to finish it. And then others have a completely separate one that's not even on this diagram. It's just a transport gene that's not associated at all. A series of transport genes. So, we looked at everything in the Thermatogae species. So there were several species in the Thermatogae family. We've got, we've got Petratoga, Cosmatoga for Vitibacter, 2 different species of Thermosipho, Thermatoga. So all of these are bacteria that are related in the same group, right? And this is presence-absence data, and it's basically telling you, so all these guys are closely related, but some of them have the genes and some don't. So you can kind of see the order that they got these genes inside of the family. So this is the transporter, and all of them have the transporter. So they could all take B12 in from the inside. Let's see, these are the scavenging genes, and some of them have the scavenging genes, but not all of them do. And then only 2 different strains of one species can actually make it from scratch. So what this data tells us is of the 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, of the 12 species, of the 12 that we knew at the time, only one of them can make it from scratch. Only a couple of them can scavenge it and finish it, and the rest of them have to get it elsewhere, right? So this is another way of showing presence-absence and the order of acquiring things from a bioinformatics standpoint. So this is kind of like looking at a phylogenetic tree with branches coming off. And at some point, basically all the things that these guys are related to, if we go back to their relatives, they have the salvaging pathway. So some of them have the salvaging pathway, but none of the ones that they've descended from actually have the de novo, the make it from scratch genes, right? So if we look back and we look at like, the red means that they can make it from scratch. And notice only this Thermosipho africanus and Thermosipho melanesiensis can make it from scratch. And then everything else can either scavenge it or has to transport it. But if we look back at these relatives, like, so to speak, the parents and the grandparents, you'll see that the organisms they came from don't have these de novo genes. So we're like, where did these come from, right? They just, these bacteria just woke up one day and they're like, sorry, Dad, I don't want to go to law school. I want to be a dancer, right? So this was really strong evidence for horizontal gene transfer. And we analyzed these genes and that represent these red de novo synthesis clusters, and we found they were really related to things in the Aquifex and the Firmicute and Archaea. So that firmly supported horizontal gene transfer. And there were further studies I'm not going to bore you with today, but we got a stronger picture of who they were acquired from. Right? So we found out that Thermosipho is the only species in this phylum that has both synthesis and salvaging clusters. Carbinamide, corinoid, and cobalamin, those are all fancy words for vitamin B12, so they can get, or pieces of vitamin B12. So we found that these clusters form a pathway that allow you to make it completely. We proved that we could make it in terms of wet lab, and we also looked at gene expression too. I could bore you for hours if you wanted to see all of that. But that was the basic, the basis of that, right? Let's see. So the carbinamide salvaging is the ancestral pathway for B12, meaning everybody before it could steal it, but it took them getting it from archaea and bacteria to be able to make it from scratch. We also found something really cool. We found out that the B12 gene clusters are regulated by riboswitches, and riboswitches are these little kind of on-off switches. And if B12 is present, it tells the riboswitch to shut the genes down, and if the B12 is absent, then it goes on and it makes more. So we even found some information about regulation of B12 synthesis. A lot of repetitive stuff, and that's my conclusion. Yay, thermocepho can produce B12, and I got a paper. So, those are 2 projects I specifically worked on. If you're interested in a career in bioinformatics, you have a lot of different options. You could be on the level of academic, a professor, either running a research lab or teaching the courses, right? You could be a private consultant. There's the money, right? You could be a private consultant that goes to work for biotech companies or even for an academic lab. You could be a medical researcher. They hire bioinformaticians by the dozens now. So it's a booming field, and I think it will still be booming for a while. I think it'll be a while before the field is saturated. So it's a good area to look into if you want to have some options. You could be a data scientist for a company where you're not associated with a hospital, but you're working for pharmaceuticals or engineering or even social media. They've got people working at Facebook that have the same skills that people that analyze genomes do, right? It's all big data. It's all churning through it. Yeah, so lots of different options there. Here's an online resource. If any of this is interesting to you, there's a website called expassy.org, and you can find tons of programs. You can find BLAST on this website. You can also find programs to analyze protein structure or genome mapping, proteins, genes, whatever you want. If you're bored on a Saturday night sometime, you just want to browse through it. And then, if none of this sold you, if anybody's had a class with me, you know I'm unhealthily obsessed with cats. And there have been some really cool recent studies using analysis of cat genomes, right? So a while ago, they basically figured out where the tailless Manx cat came from, traced its lineage. And then just a couple of days ago, a great thing, how cats conquered the world and a few Viking ships. And they did this, they looked at 200 genomes, the genomes of 200 cats that lived either between 15,000 years ago up until about 200 years ago. And they tracked migration patterns, color patterns coming from certain areas, and they found out that things like the tabby cat was a mutation that didn't show up until the Middle Ages, you know? And then they tracked that certain cats came over to North America with the Vikings and all sorts of cool stuff. So you can do anything you want with these kinds of studies. Thanks. I open it up for any questions. So informative. All your questions were answered. I love it. No. It's a lot of information, you know, big data. So, yeah, Sergio.
What's your next step in research?
My next step in research, we are expanding cataloging the B12 genes, and we're interested in looking at it across the whole tree of life. So instead of just the Thermatoga, we want to look at all bacteria and all archaea and get a bigger sense of these relationships and these patterns. And then another thing I'm interested in is some of the genes in these pathways aren't known. So, well, they don't know what the genes are, they don't know what they encode for, and they don't know what they do. So I want to go in and fill in some of the blanks. We know reductases have to be in there doing something to the cobalt metal, but we don't know what those reductases are. So I'm targeting that. Yeah?
Who designs these workstations to store these big data?
Who designs them?
The workstations, you mentioned terabytes of data.
That's a good question. I don't know. There's a lot of clusters. So a lot of people work, like a lot of individual scientists will work off of one major cluster. And usually the institute they're working at maintains the cluster. But I'd have to say I don't know enough of the wires and the bolts to tell you about that. Yeah.
Horizontal gene transfer,
Mm-hmm.
like was it observed only in microorganisms or also in higher animals?
We've seen it in higher organisms. There's a really cool instance where people in a certain Japanese population have gotten a enzyme for breaking down seaweed from their microbiome, from the bugs in their gut. So yeah, it goes further than just bacteria. And there's even some of the virus DNA problems that can also be an example. Yeah.
Any project in here that like a student can be, get into the field, like any kind of coding kind of?
I do have the project where I'm trying to look at the whole tree of life. There's probably a lot of different places. I'm starting up my program, so I don't have a ton of spots. But yeah, if anybody wanted to talk to me, I could see if there was a space for students. Yeah. Yeah. Also, if you can't find research at your individual institute, check out summer programs. There's a great program called REU, Research Experience for Undergraduates. It's funded by the NSF program. And you could find any area you're interested in. If it's bioinformatics or maybe it's organic synthesis, you could find an REU program, go there, and they'll actually pay you for a few months to work at a university and learn stuff. Cool. Thanks. I'll be sticking around a bit if anybody has any specific questions. Thank you for your time.
Thank you.
Yeah. What's up? Thanks. Thanks for coming. Hi. Yeah, thank you very much.
It's the easiest one, just to give a minute, vaccine, and then the next age of it, and then the
Right. Yeah, you can track resistance. with this kind of stuff.
resistance, exactly.
Yeah.
Is that because of horizontal gene? But not for me,
It could be. I think some of it might be horizontal gene transfer, but a lot of it is probably just survival of the fittest. Yeah. So it might be a little bit of both. Yeah, good point.
You should. Yeah.
We have it here in this classroom in which competition in the morning,
Company makes. I work for this company, and
Okay.
they what they do is they create a method of inhale insulin to your lungs,
Oh, okay. Yeah.
and they come up with a
I've heard of the oral form.
and I have some example of the way that they do it.
Okay.
Here it's like you get like a little
Inhaler?
inhaler.
Okay. Very cool.
And yeah, it's very. What happened was that they what they do is they have these cartridges with certain amount of